Hello everyone, my name is Luis Brito and I'm an assistant professor at Purdue University in the United States and an adjunct faculty at the University of Guelph in Canada. And in the next 12 minutes, I will briefly describe the work that we have done as part of the Working Package 6 of the SMARTER project on the characterization of genetic evaluation systems and breeding programs in sheep and goats. I also want to thank all my co-authors for their contributions in this work. So there are more than 700 sheep and goat breeds raised worldwide, summing up to a total population of approximately 2.3 billion individuals. Despite the large population size, genetic improvement has lagged behind compared to other livestock species, such as dairy cattle, pigs, and poultry. And breeding programs have been implemented only for a limited number of breeds in the case of small ruminants. So some countries, such as France and the United Kingdom, have developed sheep and goat breeding programs over the past few decades, which have indicated the great potential of genetic selection in these species. Substantial genetic progress has been observed, so as shown on these figures for two French meat sheep breeds for prolificacy, maternal ability, and growth. And in addition to this clear genetic progress over time, the fact that there are similar breeds being raised in different countries is an opportunity to develop collaborative research and cross-country genetic and genomic evaluations to enable the optimization of breeding programs, especially in the case of smaller breeds, and also to maximize the genetic progress for larger breeds that are raised in multiple countries. Considering the large diversity of sheep and goat breeding programs, gathering knowledge on the alternative approaches that have been taken in different countries is paramount to the design of efficient and integrated genomic breeding programs. Therefore, the main goal of this activity was to describe the genetic and genomic resources that are available, the genetic evaluation systems, and the breeding schemes of sheep and goats in countries that are participating in the SMARTER project. This was done through surveys that were developed and distributed to all partners, and it contained 59 parameters about general information and population description, general evaluation systems, and genomic evaluation at both the research and implementation stages. And additional information was obtained from published papers, from project reports and evaluations, and field visits as well. And this was done for the three main breed groups of meat sheep, dairy sheep, and dairy goats. So we summarized information from 48 breeding programs ongoing in 12 countries: France, Greece, Hungary, Ireland, Italy, Norway, Romania, Spain, Switzerland, the UK, Canada, and Uruguay, which included information for multiple breeds and more than 3 million animals involved in data collection schemes. With regards to the genetic resources, for dairy goats, the most common breeds are Alpine and Saanen, or composite breeds involving these two breeds, such as Norwegian dairy goats and Yorkshire goats in the United Kingdom. However, there are other breeds, such as Toggenburg in some countries, and local breeds such as Alba de Banat and Carpatina in Romania. A larger number of dairy sheep breeds are involved in genetic evaluations, and the Lacan breed which is a French breed, is present in most countries. There is a much larger diversity in meat or dual-purpose sheep, but there are also some common breeds, such as Texel, Charolais, Suffolk, and Vendée. The main group of traits recorded across countries are, in the case of dairy sheep and dairy goats, milk yield and milk composition, indicators of mastitis, udder and body conformation, and reproduction. With regards to meat sheep, there are mainly growth, reproduction, health traits such as lameness and mastitis, ultrasound, wool in the case of dual-purpose populations, and carcass traits in which some of them are ultrasound-based. However, novel traits are also being investigated, such as feed efficiency, methane emission, and behavioral indicators. And there are some countries that do have central test stations in which additional phenotypes are also being collected. Progeny test schemes are still ongoing, especially for dairy sheep and dairy goats, and both the number of progeny tested males and the target number of daughters per male vary across countries, going from 5 to 150. Pedigree completeness is a very important parameter for accurate genetic evaluations, and we found that the average proportion of animals with unknown parents is around 20%, and also the average generation interval is 4.5 years for dairy sheep, and 4.2 for dairy goats, and 3 years for meat sheep. With regards to genetic evaluations, there is a large variability in data recording and use protocols, such as recording only on the first parity or multiple parities, data collected by producers or by trained technicians, or a combination of both. And with regarding to data inclusion, there are data that has been included since or before the 80s for some breeds in France and Canada, but for the main traits, they have been recorded since 2000 in almost all partners. Reasonably similar heritability estimates have been observed for similar traits across countries. And there is also a large variability in the statistical models used, such as single or multiple trait analysis, single or multi-breed evaluations, as well as the number of evaluations per year that range from 1 to 24. The models that are used, such in the case of the dairy species, for example, multiple trait repeatability or multiple trait fixed regression test day model, or multiple trait random regression models in the case of some partners. So here is just to indicate the variability on the number of genetic evaluations that are reported to producers throughout the year, and it does depend on the species as well as in the country. The breeding values are also expressed in different units or scale, which can be from the original scale with a fixed base, with the original units, or in genetic standard deviations, but scaled to a certain mean and standard deviation, and so on. So there is some variability on how the genetic breeding values the breeding values are presented to the producers. With regarding to genomic evaluations, some countries have already implemented genomic selection for some breeds, and others are working on the development of reference populations. And there are currently over 40,000 genotyped animals. These animals are being genotyped based on similar panels across countries in some cases, but in other cases there are greater variability, especially in the case of meat sheep, in which more than 3 different panels have been used across countries. So, as final considerations, there are numerous challenges to be addressed, such as the disparity of trait recording across countries, the different SNP panels, different statistical models, and different evaluations. So there is a lot of variability. However, there are many opportunities to use the resources that have already been developed and are in use in some of the countries to work collaboratively to optimize selection for novel breeding goals such as resilience and efficiency in small ruminants across countries. And all the detailed information will be provided in a scientific paper, which is in preparation. And with that, I would like to thank all the smarter partners and you for the attention, and I would be happy to take any questions from the audience. Thank you very much.